but I can't see you right now. I cannot see you.
Okay. Actually, connection is the issue today here.
Okay. Very good morning to you all. And today we will be joined by Dr. Anant Narayan Bhatt. So I take this opportunity to welcome Dr. Anant. And Dr. Anant is a senior scientist at DRDO. And let me introduce Dr. Anant. So can you see the slides, sir?
I can't see currently.
Okay. So welcome to our platform, which is called Knowledgians, where head meet hearts. And today's topic will be on 2DG, an anti-COVID drug, from an age-old molecule to a modern antiviral therapy. And we are going to talk about 2DG, COVID ki Sanjeevani. So, and the focus of today's lecture series is drug discovery. And we know कि कोई भी जो वैज्ञानिक होता है, every scientist and the life of scientist is full of ups and downs. And apart from adding the basic understanding in science, the dream of every scientist is to add value and ease the suffering or make life better. So आज हमारे साथ Dr. Anant Narayan Bhatt होंगे, who is working as a senior scientist at DRDO, and he will be sharing his knowledge and experience and telling the story behind India's own attempt of repurposing 2DG for antiviral therapy. So we together dedicate this talk to two great scientists. One is our own mentor, Dr. Vinod Bhakuni, who has mentored both of us. And it has been a privilege to have Anant sir as my senior in the same lab. And the other is a common friend of ours whom we lost. They are no more. We lost Dr. Bhakuni in 2011. And in 2018, we lost another important scientist who has been instrumental in shaping my thought process and also a very close friend of Anant sir, Dr. Manish Goswami, who was a scientist at Park. So before I introduce Anant sir, I would like to take the quote from APJ Abdul Kalam, where he says that I am not handsome, but I can give my hand to someone who need help, because beauty is required in the heart, not in face. कि सौंदर्य आत्मा की है विषय वस्तु, समझ जो ये गए फिर जीवन तथास्तु, खूबसूरत है वही जो किसी के काम आ सके, किसी के जख्म घाव पर मरहम लगा सके, जादू की झप्पी दे सके निष्काम भाव से, पीड़ा का हरण कर सके त्वरित प्रभाव से. और ऐसे ही एक वैज्ञानिक जो हमारे साथ हैं, डॉक्टर अनंत नारायण भट्ट, जिन्होंने जो ये विपदा का समय था, ऐसे में he has worked on a very important molecule, and that has definitely helped easing the suffering of the people throughout Pan India. So welcome, sir, and I invite you to enlighten us on the topic 2DG for antiviral therapy. Anand, sir. Hello.
Thank you, Ranjit, for introducing me. Can you hear me?
Yeah, I can hear you.
Okay. Is my slides visible?
Not yet.
Now?
Not yet, sir.
Okay. Actually, there are some internet issue because of the rain yesterday,
Yeah,
heavy
yeah.
rain in Delhi.
Yeah, but I think you can talk in general, and then when it feels like you
Okay.
can. Yeah, now I think the sharing is starting. Yes.
Okay. Now is it there?
Not yet. It says
Yeah.
that it started sharing. Yes.
Just share it again.
Yeah. Yeah. Now I can see. Sir.
You can see. Okay.
Yeah. Very nice.
Okay. So is it in presentation mode now?
Yeah, it's in presentation mode. Yes.
Okay. So as Ranjit introduced that today we are going to talk about 2DG as an anti-COVID-19 therapeutic, and from an age-old molecule to a modern antiviral therapy. So we are going to talk about this in purview of how this molecule has evolved and how we repurposed it for as an anti-COVID therapy. So as Ranjit already introduced, that we come from a kind of same lab, and both were mentored by Dr. Vinod Bhakuni, who lost his life in very early age. I mean, in 50s itself, in 50s, and he was a great mentor. He was a great person, and it will not be I mean exaggeration if I say wherever I stand today is because of him. So he was our PhD supervisor, and Manish is another friend who lost his life to cancer just when he touched 40s. So I mean, he was a very good friend, Manish Goswami, and he was my MSc classmate, and a person who was like highly enlightened with science, and who always wanted to pursue science, wanted to do something good in this area. So I just want to tribute my talk to these two people. And when Ranjit contacted me, he also said that probably we should remember these two people who were part of our scientific journey from the beginning. So so I just want to tribute and dedicate this talk to these two people. So just going before going to this topic, I just want to remember one scientist long back in 1920s and 30s, Dr. Otto Warburg. He was a German physician, and then he said, cancer above all other disease has countless secondary causes, but even for cancer there is only one primary cause, and that cause is the aerobic glycolysis, which is known as Warburg effect today. So he said that the prime cause of cancer is the replacement of respiration of oxygen in normal body cells by fermentation of sugar. Now, when we understand cancer more and more currently, we know that it is always a metabolic plasticity of the cancer cells, which keeps switching between high glycolysis to high mitochondrial respiration in different stages when we talk about heterogeneity of cancer cells. But it was his primary observation, and based on that, he got Nobel Prize for this work. And not only he, there was a later scientist, Louis Sokolov from US. He also got Nobel Prize for based on his hypothesis that cancer cells take a lot of glucose. He designed a molecule called FDG, fluorodeoxyglucose, which is used commonly for PET scan. So that is a diagnosis technique which is used highly for cancer. and that Louis Sokolov also got Nobel Prize for that invention. So I just want to remember Warburg, who gave this concept in 1930s, and he published this work in Science. And based on his work, actually, when he said that the glycolysis is one of the predominant pathway which is increased so much in cancer cells, there are so many glucose analogs which were designed to inhibit the glycolysis. 2-DG is just one of them, 2-deoxy-D-glucose. And you can see the difference between glucose and 2-deoxy-D-glucose is just one. That is, at position 2, it is just replaced by oxygen, or hydroxy group is just replaced by hydrogen. So there is a lack of oxygen there. And because of this lack, it gets internalized, outside the cell. When it circulates in the body, it mimics exactly like glucose. Body thinks that it is just simply glucose. So body doesn't react to it. And this keeps moving in all around the circulation, just like glucose. So there is no problem, no side effect. But when it internalizes in the cell, what happens? It gets converted into 2-DG6-phosphate very easily, and it gets trapped in the cell. And once it gets trapped in the cell, it is not a good substrate for the next step of the glycolysis. So it does not get metabolized. It does not give energy. It does not give any further benefit later on. So that is why this is a anti-metabolite. It's called glucose anti-metabolite, and it inside the cell, it works exactly opposite to glucose, and then it cannot translate the energy there in the form of energy or anabolic intermediates. So that is why the interest in cancer biology, in virology and several other things, where glucose is the predominant and primary demand of the cell, goes very high for this molecule. So just to tell briefly that how basically it happens in cancer cell, on top you can see that normal cells have normal glucose demand. Briefly that how basically it happens in cancer cell. Hello?
Yes, sir. You can continue. Yeah.
Okay. So in normal cell division, you can see like when the cells are progressing with normal pace, they don't need too much glucose. So their demand for glucose is very minimal. And in the cancer cells, what happens, like when they produce lot of, they need the cell proliferation has to go at very fast pace, at a very rapid pace, then the glucose demand goes significantly high in these cells. And you can see, that is why if 2-DG is given, it can stop the fast proliferation of these cells. And fast proliferation of these cells can stop the tumor growth. But when the tumor, like when the patient comes to the clinic, he is with the fully blown tumor, right? So then you need to increase the tumor, you need to decrease the tumor burden. You need to kill the tumor cells. So in that, 2-DG alone is not sufficient, and that is why, because it can stop the tumor cell proliferation, but it cannot kill the tumor cells. And if it is at all at some concentration, it is able to kill the tumor cells also because of the oxidative stress generated by metabolic oxidative stress generated by it. Then also it is not that efficient in that, and that is why you need to combine it with chemotherapy and radiotherapy. So this is the basis why it was used a lot in cancer biology. And literature is full of use of 2-DG in cancer as an anti-cancer molecule. So with this background, long back, one of our former director, Dr. Vinay Jain, he brought this molecule. He did his PhD from Germany, and he worked with some of the lineage of Dr. Warburg. Dr. Poholit, he worked with Dr. Poholit, where he was using this concept of targeting cancer cells. So he brought this molecule to India, and long back he worked in NIMHANS, he worked in AIIMS as faculty, and then later he joined as our director at INMAS DRDO long back in 1996 or even before that. So they worked thoroughly on this molecule for its cancer effect. And then they started clinical trial of this molecule in cancer patients, mainly brain tumor patients. So these clinical trials were conducted at AIIMS Delhi. Tata Memorial Hospital, Mumbai, Kidwai Memorial Hospital, Bangalore, Nizam's Institute of Medical Sciences, Hyderabad, and Dharmshala Cancer Hospital, Noida. So they did a extensive clinical trial of this phase 1 clinical trial, phase 2 clinical trial in independently in brain tumor patients. And then what happened, like in 1998, this is a beautiful story that after this Pokhran blast, everybody knows that US put a lot of sanctions on India. I mean, they stopped exporting a lot of things to India. So 2DG that time, because clinical trial was going on in 1998, 2DG was also one of them. We were buying 2DG that time from USA. So 2DG sanction was also given because it was a defense lab, and defense lab was buying that. Defense lab was mainly involved in the testing, Pokhran testing. So they stopped selling many things to defense labs. 2DG was also one of them. So that was the time one of our chemistry lab that's called DRDO Gwalior, Defense Research and Development Establishment. It is situated in Gwalior. So that lab has expertise in chemistry. So we contacted them, and then they synthesized it for us, the 2DG by another method, and that method is also got patented by us, DRD Gwalior and INMAS together in India. And that method actually that technology we transferred to Dr. Reddy's lab in 2004. So after transferring that technology to Dr. Reddy's lab in 2004, we took their help in conducting the phase 3 clinical trial in brain tumor patients. So from 2004 onwards, this phase 3 clinical trial in brain tumor patients started. And later in 2014, DCGI recommended this drug for use and for marketing and production and marketing authorization to Dr. Reddy's lab in 2014 for cancer patients. So this is the brief history from 1996 to 2014 about the clinical trial of this drug in brain tumor patients. So what is the rationale behind using 2DG as an antiviral drug then? So, like as I said, cancer cells are also very fast proliferating, and anything which proliferate very fast or when it goes in emergency, the natural evolution of the cell or the organism is like that. It does not, we cannot supply the oxygen at the level of the requirement of the cells. So that is why like when we are running, when we are fighting, like adrenaline when secretes 3F hormone. So fight, flight, and fright, all these conditions secrete lots of adrenaline. And the primary job of adrenaline, it does many things, but the first thing what it does, it stops the mitochondrial respiration. So the moment mitochondrial respiration is stopped, that is the time when you are fighting, when you are running, when you are running from any situation or anything, that is the time need maximum energy, right? And from where this energy will come? So this energy comes from glycolysis. And like glycolysis is efficiently very it is not an efficient pathway. It is efficient in the sense economic, sorry, it is not an economic pathway. That means it takes one molecule of glucose and just give 2 molecules of ATP, right? However, when the glucose goes through the entire oxidative phosphorylation, then it produces, depending on the shuttle, whatever it is using, 32 or 36 molecule of ATP, right? So it is not an economic pathway, but the thing is that its rate is very rapid. So because of its very rapid rate, the energy production in very minimal time can be very high as compared to when the glucose is getting metabolized through complete. So that is why the glycolysis is a preferred pathway during emergency, during this kind of situation. So anything which proliferates very fast needs glycolysis, support of glycolysis in the host cell or in the cell. So in case of virus, it is a host cell of human body. So upon infection, the virus hijacks and reprograms the host cell metabolism. So you can go and check in the literature, whenever what happens, like whenever viral infection happens, it damages the mitochondria significantly. So what happens? A lot of calcium disturbance happens, and that calcium gets accumulated into the mitochondria mitochondria, because of which mitochondrial function gets compromised. So what happens, like once mitochondrial function gets compromised, the cells and mitochondrial oxidative phosphorylation and so many other things cannot happen in those cells. So as a defense mechanism or as a compensatory mechanism, cells increase their glycolysis. So this is what happens. So virus has the complete regulation of this reprogramming. So the moment virus infects the host cell, it reprograms the metabolism to increase the glycolysis. And there is ample evidence in the literature for this rationale for using. So this is one thing. And then why it needs. So energy is one thing. Then the second thing, the enhanced demand of virus infected host cell. See, virus, when entered in the cell, it just was one copy, but now it has to multiply, it has to produce several hundreds or thousands of copies there. So for that, what it needs lipid, it needs nucleic acid, it needs protein for production of a newer viruses, right? And it needs, of course, the glycans also, because majority of the virus proteins are glycosylated. So once those proteins will be glycosylated, so it needs all these basic raw material to synthesize new virions. So what will happen, like once glycolysis is inhibited, majority of the glycolytic intermediates are contributing to these basic pathways which are responsible for a lot of biochemical pathways which are responsible for synthesizing this new molecule, nucleotide, triple P pathway, and so many other things. So glucose is basic. Glucose and glutamine are the basic source for these for synthesis of new viruses. So these are anabolic requirements also, which does not get fulfilled if you inhibit glycolysis. Then while oxidative phosphorylation provide this, I already talked about that ATP rate production by using glycolysis is much higher. I mean, if I just give a approx estimate, like in 1 minute, if suppose that one molecule of glucose goes through complete glycolysis and mitochondrial respiration and produce just 36 ATP, if glycolysis has gone significantly up, what will happen? 50 molecule of glucose will be converted during the 1 minute time. So overall output would be 100 ATP. 2 molecule of ATP from every glucose. So 32 or 36 versus 100 molecule of ATP will be produced, and that is why glycolysis is the preferred pathway in during this kind of emergencies. So and unlike normal cells, like normal cells has this plasticity. If you stop its glycolysis, it will get energy and other things from mitochondrial respiration. But virally infected cells, because oxidative phosphorylation or mitochondria is already compromised in them, so they are relying only on glycolysis. So if you inhibit glycolysis in those cells, their metabolism will be completely choked as compared to normal cells. So this is a very beautiful differential metabolic requirement of virally infected cells, which can be targeted using 2DG. So this is a brief diagram. You can see here that on the left side, this is a normal cell. Glucose is entering and producing pyruvate, and that goes to the mitochondria, and then lipogenesis and so many other things are happening normally. And the expression of GLUT transporters are also much less. Those which are responsible for transferring glucose is much less in normal cells. However, in virally infected cells, what happens? Like once the virus infects this cell, as I said, it reprograms the metabolism. So that reprogramming involves what? Lipogenesis or mitochondrial respiration will go bare minimum, and then lot of production of GLUT transporters will happen. So on the surface, there will be a lot of accumulation of GLUT1, GLUT4, and recently we found in our lab that GLUT3 is also one of the glucose transporters, which starts expressing on the surface of cell. So GLUT3 has the lowest Km value. That means even if there is a scarcity of glucose, the requirement of glucose of the virally infected cells will be fulfilled, because its Km value is in the range of 1 millimolar. And you know, like the normal circulating level of glucose in our body is around 5 millimolar. right? 5.5 millimolar. So even at very low concentration of glucose also, virally infected cells, they do get glucose, and their multiplication cannot be stopped, right? So what happens, like when you give 2DG, so what will happen, that will also get selectively accumulated in virally infected cells. Why? Because they have very high amount of transporters, glue transporters on their cell surface. So this is a beautiful mechanism of this drug, that its accumulation happens selectively in virally infected cells, right? Then after that, we know, I mean, we discussed a lot about how it inhibits the energy production, because in glycolysis, after step 1, it cannot process next steps, and that is why it will be stopped. So energy production will be stopped. Then anabolic intermediates which are coming, like for nucleotide biosynthesis, for lipid biosynthesis, those will also be stopped. So those will also stop. And then the third mechanism, which we will see the experimental evidences also for that, is I said key all these virus proteins are basically glycosylated, spike protein mainly when we are talking about SARS-CoV-2. So these proteins, they take a lot of glycans. They are glycosylated. So mannose is one of the primary requirement for them. So like 2DG exactly mimics to mannose also. Mannose is a glucose epimer, and 2DG mimics exactly like mannose, because at position 2, this is a just hydrogen now, and in mannose also the lower position is hydrogen only. So it mimics exactly like mannose, and cell gets confused. It incorporates mannose into 2DG into in place of mannose. So once it gets incorporated, the extensions which are happening at position 2 because of hydroxy position, that hydroxy is not there in mannose. So what will happen in 2DG? So that termination, premature termination will happen in this, and there will be misglycosylation. So because of this misglycosylation, it is like that you are writing a letter, and you just write a wrong address on the letter. So what will happen? Letter will never reach to the right place. So this is what happens with the progeny virions. So what happens, like these progeny virions, they have misglycosylated spike protein. So because of that, they cannot go and infect the new neighboring cells, right? So so this has a beautiful mechanism, and we will test these mechanisms one by one experimentally in our lab. So you can see here on the left side, this is a model, and you can see like in this lung epithelial cells, you can check these are the ciliated cells. There are only 2 cells which are virally infected, and this is a model which shows that only viral infected cells accumulate lot of 2DG, not the normal cells, right? Those cells which are not infected, they are not accumulating, and this is the experimental validation of it. On the top, you can see that the glucose uptake has gone significantly high in virally infected cells, but in the lower panel, you can see the image which comes from this is a qualitative validation. The left side, on the left side, we have stained the cells with the cell tracker red. This is the cell which gets this doesn't take infection very high, viro cells, which is exactly similar to viro E6. Viro E6 takes a lot of infection, but viro cells are taking they take very minimal infection, or they don't take, good population of it doesn't take infection. So so we colored them with red, and we co-cultured them, and we reinfected these cells. We infected these cells. So you can see that the red colored cells, they have taken very minimal, this is green color is coming from 2-NBDG fluorescence, which is a glucose analog, fluorescent glucose analog. But you can see those cells which are not stained in red, they have taken very high amount of glucose. So that means the infected cells are basically taking very high amount of glucose. So this is evidence coming from when we will give this drug to humans, there will be mixed population of infected and uninfected cells in any tissue. So this will selectively go and get accumulated into virally infected cells only, and not in uninfected cells. Or in uninfected cells, if there will be accumulation, there will be very minimal accumulation. And as I said, in normal cells, the metabolism is taken care by both glycolysis and oxidative phosphorylation. However, in infected cells, the metabolism is taken care only by glycolysis, not by oxidative phosphorylation, predominantly by glycolysis, not by oxidative phosphorylation. So that was one evidence that its accumulation is very selective in cells. Then the second evidence comes from this experiment where we conducted this in supernatant and cell-bound. This was the primary experiment based on which we got the clinical trial approval. This experiment was conducted at CCMB Hyderabad. The previous experiment was conducted in my own lab, BSL3 lab here at INMAS. And you can see here that this inhibition of viral multiplication, which we monitored by using, estimating two different genes of SARS-CoV-2, E gene and RDIP2 gene. And you can see that at very low concentration, which can be easily achievable by giving this drug orally, IC50 concentration 1 millimolar, you can see that inhibition is taking place. And in cell bound, it is even lower, like 0.6 millimolar only. The inhibition of viral multiplication can be obtained using 2DG. Then we tested the second hypothesis, what we were telling that due to misglycosylation, what will happen that the cells will not be able to infect the neighboring cells. So you can see here that what we did, we treated the cells with we infected and then treated the cells with drug 1 millimolar and 5 millimolar 2DG, and that will definitely reduce the viral copy number as we have seen in previous experiment. So what we did, we normalized the viral concentration and reinfected fresh cells with these viral progenies. And after reinfection, you can see with the equal number of viral progenies, you can see the viral infection has gone significantly down in the from the progenies which were taken from 1 millimolar and 5 millimolar treated 2DG. So what it suggests, that the virions which were produced, they were not able to reinfect the newer neighboring cells. Then a lot of people express this concern that because it was used in cancer cells, and people thought that because it kills cancer cells, it will kill our own virally infected cells. But the idea is not to kill virally infected cells. The idea is to stop the virus multiplication in the cells. So this data, which is coming from virus-induced cytopathic effect or apoptosis, you can see here that the middle row, which takes the viral infection, the red color is very high, which is coming from ethidium bromide. The uptake of ethidium bromide in dead cells like apoptotic, late apoptotic and necrotic cells are very high. So because of that, there are a lot of dead cells. But when you treat this infected cells with 2DG, you can see that the dead cell population is not that high. That has gone significantly low. And these are this right side is the derived information from these images after this image analysis. And you can see that the uptake of ethidium bromide is significantly low as compared to virally infected cells. So basically, what it is doing, that it is not letting the cells to die. Viral multiplication goes significantly down, and because of that, the death is not occurring. It is not that 2DG is helping in survival of those cells. Basically, 2DG is not letting the virus multiplication happen, and that is why it is not the cells are not dying in 2DG plus infection group. And this cytokine storm and so many things are happening. The two predominant reason is for that is why, because the lung infection, the pneumonic lung infection which is happening, that is one thing. And then second thing, lot of cell death which is happening, our own cell death, lung cell death which is happening, that all the cellular debris, they cause cytokine storm. So once cytokine storm will be there, body is not able to come out of it, and that is why a lot of deaths are happening in COVID patients where this cytokine storm is happening. So if the cell death of the virally infected cells will go significantly down. You know, like by this mechanism also, it can control the the inflammation as well. So this is the overall summary of what we have been talking. Like in the top panel, you can see that SARS-CoV-2 infection is happening in first infected cells, and you can see like the replication is going on. It is releasing the virions, which is infecting the neighboring cells, and neighboring cells are dying, and because of that death, inflammation and other consequent consequently effects are happening. But in case of lower panel, you can see where we have given 2DG, what happened, like the energy production is stopped, anabolic reactions are stopped inside the cell in virally infected cells where 2DG got accumulated, and it enhanced the misglycosylation. So because of this, the production of the virions is very less. Then the second thing, whatever virions are forming, they are defected virions assembly. So those virions are basically defective virions, and those defective virions basically are not able to cause the infection in neighboring cells. And when they are not able to cause infection in neighboring cells, the cells are not dying, right? So because of this region, we are not seeing too much inflammation in that. So this was the antiviral effect of 2DG. Now 2DG is very well established anti-inflammatory molecule also. Why? Because like as we said that for glucose is the basic requirement of cancer cells, glucose is the basic requirement of this what you call virally infected cells. Glucose is similarly the basic requirement of inflammatory cells as well, because inflammation cannot happen by without inducing glycolysis. So if you inhibit glycolysis, inflammation cannot happen. This is very well established phenomena in literature. You can check this. So what people did, people caused lung damage by infecting or instillating the LPS directly into the lungs. So what happened, you can see the plasma IL-6 level has gone significantly high. In the bar diagram, you can see the plasma IL-6 level has gone significantly high. And once you give 2DG, the plasma IL-6 level goes much down, right? So it can control the inflammation. That is a very clear evidence. And then pulmonary IL-6 level also, you can see this was estimated in BAL fluid. So in BAL fluid also, in the bronchoalveolar lavage of the lungs, there also the IL-6 level has gone significantly down. And not only this, you can see like by treating 2DG with 2DG, the mortality rate or LPS-induced mortality rate of mice has also gone significantly down. So this is a very clear evidence that 2DG can regulate the inflammatory damage which is happening in the lungs also. And this evidence is recently published in 2018 in PNAs. And this evidence is also coming from rhinovirus infection. So rhinovirus infection also is a respiratory infection that takes place in the lungs and then what you call this throat and other places. So there also when 2DG was given, you can see that all the inflammatory markers has gone significantly down. And the histology of the lung also shows that the inflammation is not seen in the mice lung when they were treated with 2DG. Other than this, 2DG is very well known, you know, being an anti-metabolite, anything which is proliferating very fast, like all the microbes. So even if it is not happening in the cell, because they are cellular organisms, so the 2DG accumulation will happen there also, and it will stop the fast proliferation. So similarly in yeast also it has been shown. And you must be knowing, like in India, other than 2DG, like all the COVID patients, they are picking a lot of this mucormycosis, which is another fungal disease. They are picking a lot of infection there. So we are conducting this experiment whether 2DG can inhibit the mucormycosis growth also in COVID COVID patients. But whatever data we already have, that suggests that it can work very well in fungus also, to counter the growth of fungal cells also. So, and this is the data from phase 3 clinical trial. You can see here that in 2DG plus SOC group, almost similar patients came with yeast infection when they were recruited, and when they were discharged, in the end, you can see like only 6 patients, half number of patients had microbial infection, and those yeast infection has gone significantly low. I mean, there was hardly any infection was found in those 6 patients, average infection. However, in a standard of care group, standard of care means here in a standard of care, all the antivirals were given. So in a standard of care group, it has gone further up. It has not gone down. So this shows that it is a very good. It can be a very good anti-fungal activity. Can also be seen in this molecule. However, we are conducting experiment. Until we conduct the experiment specifically in mucormycosis, we don't want to comment on that. So phase 2 and phase 3. Based on these results, we got approval from DCGI to conduct phase 2 clinical trial in May 2020. And later, then we did dose ranging. In dose ranging, we, conducted the trial on 2 higher doses as well. And we selected a best dose at which we are getting best efficacy. And with that dose, we had gone in November 2020, we have gone for phase 3 clinical trial. And we got approval from DCGI to conduct phase 3 clinical trial. And with that, that was conducted up to March. In April, we analyzed the results of interim analysis, and then we presented to DCGI. And on 1st May, we got the approval for emergency use authorization. So the main dose which we selected in clinical trial is 90 mg per kg per day. This is equally divided in 45 mg per kg per time in the morning and AM dose, and 45 mg per kg PM dose. So it is divided in 2 equal doses. And then standard of care, this was given on top of a standard of care. And in standard of care, like all the whatever is ICMR protocol and recommended antivirals and all, they were completely given. And this was given on top of it. So when it was given on top of it, we found that in patients, we got median 2 to 4 days relief on various endpoints. Like that there are a lot of buzz about this. This is giving very good relief from supplemental oxygen dependence. So it decreases the requirement of supplemental oxygen very fast. So in 3 to 4 days, people are coming out of oxygen. In every 2 days, after approval, we have given to a lot of patients who were on ventilator. They were critically ill. So when this drug was given, in 2 days, they came out of from ventilator to high flow, and from high flow to from BiPAP to high flow, and then from high flow to low flow of oxygen. So every 2 days they came out of it. And then in those patients who were very critical, they came in average, if we see the time, then in 6 to 10 days they came out of oxygen. So we are getting very good response in patients after approval also when we have given. And based on these results, DCGI has given approval to us. And these are the brief thing how and where it can be used. So 2DG is approved for emergency use as adjunct therapy to standard of care in the treatment of COVID patients in the hospital settings. And then ideally 2DG should be prescribed as early as possible by doctors for moderate to severe COVID-19 patients for maximum duration up to 10 days. And we in our clinical trial, we did not recruited uncontrolled diabetic patients and patients with severe cardiac problem and ARDS, severe ARDS, because mild ARDS they were recruited. And then severe hepatic and renal impairment patients have not been studied yet with 2DG. Hence, caution should be exercised when doctors give this drug to such patients. And 2DG should not be given to pregnant and lactating women and below 18 years, because trials were not conducted on these kind of subjects. So this entire exercise of last 1 1 year or 13, 14 months, it came in the form of this small sachet. And this small sachet can be dissolved in half glass of water, and people can easily drink it. I mean, it is very easy to use as compared to other drugs. And uh, this that's how the simplicity of the molecule, that's how the simplicity of this drug. So I mean, in the end, I would like to acknowledge my chairman, who was stood behind this concept, behind this entire journey. And like in April 2020, when there was a nationwide lockdown, you cannot move out. That was the time I was sent by a special flight to conduct this anti-SARS-CoV-2 activity to Hyderabad, CCMB Hyderabad, where I conducted this study. And then we found that with the help of CCMB Hyderabad, and we found that this molecule is active against SARS-CoV-2. So it was only possible in DRDO. And I mean, in the beginning, a lot of experts were telling that I don't know whether this molecule will be successful or not. So that was the time when Dr. Reddy said that if there is even 0.1% probability, let's try this concept, and let's try if whether it works or not. And that was the hope, that was the trust which drove us to this entire journey of more than a year. And we are able to we were able to translate this molecule. And my thanks and a lot of credit goes to Dr. Vinay Jain, who brought this molecule to this country. And he did enormous research, conducted clinical trials on cancer patients. Dr. Dwarkanath also, who was his associate, Dr. Dwarkanath is his student, Dr. Vinay Jain's his student, and he is my mentor. Actually, Dr. Jain only, I mean, I was graduated in protein folding and protein engineering. So when I joined in MAS, he is the man who taught me metabolism. He taught me what is its role in cancer biology. He is the man who mentored me at MAS. And I mean, this credit goes to him because of the knowledge which he gave to me. And then Dr. Channa, who is my head, and he was also one of the senior team member of my group who worked on this molecule. And all this effort, what we are seeing is coming from him also. He also contributed a lot to this program. Then Abhishek, Yogesh, these are the two people who are working relentlessly, tirelessly. And since last several months, actually, they have not taken any leave. I mean, even on Saturdays, Sundays, they're continuously working on this program. And Neeraj is the person who designed all these pathways. I mean, the figures, the graphics, how this works. So she is the person who designed all these things. And beside this, there is, I mean, it is difficult to mention. There is one scientist with us, Dr. Vijay Kumar, who helped us a lot. I mean, this clinical trial data, every one patient data is kind of one thesis. So when we did 220 patients in phase 3 and 110 patients in phase 2, you can see like we analyze almost 330 theses. So to reach to any conclusion. So he was the man who helped us a lot in taking out all the data from the electronic sources. And then we used to analyze with his help. And he helped us a lot in this. So there are a lot of people behind this in MAS also, and in DRD Gwalior also, who synthesized this. So thanks to all this in MAS and DRD team. And there are a lot of people in Dr. Reddy's lab also, sitting in India and in US, his team. And then they also work day and night, Dr. Sagar, Dr. Shenoy, Dr. Mallikarjun Rao from Dr. Reddy's lab. So all these people, they contributed a lot to this program. And because of, lot of these things, only it could successful. एक हिंदी में कहावत है कि अकेला चना भाड़ नहीं फोड़ता है. तो ये ढेर सारे चने थे जिन्होंने मिलके शायद ये जो एक भारतीय मॉलिक्यूल, एक ड्रग था
जी।
जो भारतीय ड्रग है, मतलब मॉलिक्यूल तो वेल नोन है पूरे विश्व में, लेकिन मतलब एक एक पहली ड्रग है जो इससे पहले, जैसे आप सभी जानते हैं, रेमडेसिविर, फेविपिराविर दूसरे वायरल डिजीजेस के लिए अप्रूव्ड ड्रग्स थी, तो
जी।
उनको बहुत आसान था सीधे फेज 3 में लेके जाना, लेकिन हमारे लिए आसान नहीं था क्योंकि ये एक अप्रूव्ड ड्रग नहीं थी, और ये एक मॉलिक्यूल से हमने इसको एक ड्रग का अप्रूवल दिलाया है, और ये पूरी प्रक्रिया जो हुई है ये भारत में हुई है। तो मतलब एक मॉलिक्यूल से लेके इसको ड्रग तक लेके जाने की जो पूरी प्रक्रिया है, ये पूरा डेवलपमेंट भारत में हुआ है। तो ये पूर्ण रूप से एक भारतीय ड्रग हम इसको कह सकते हैं। इसका पूरा डेवलपमेंट जो एंटी कोविड ड्रग के रूप में हुआ है वो पूर्ण रूप से भारतीय है, वो पूरा प्रयास, और लोगों को इससे लाभ मिल रहा है। We are getting lot of messages from the patients that I mean, it gives the immense happiness when they say that you saved the life of my husband or my wife or my family member. So that gives immense happiness कि लोगों को विश्वास भी हो रहा है इसमें, और ये दवा लोगों को लाभ दे रही है। इसी के साथ बहुत बहुत धन्यवाद, और
धन्यवाद
अगर
सर,
कोई
आपने
प्रश्न होगा तो जी।
धन्यवाद सर, आपने बहुत ही अच्छे से एक्सप्लेन किया है। और मुझे नहीं लगता है कि जो विज्ञान के समझ रखते होंगे उनको दिक्कत हुई होगी समझने में, कि आपने बहुत ही मतलब ये एकेडमिक लग रहा था। एक लेक्चर जो है काफी मतलब स्लाइड बाय स्लाइड, हम लोग ऐसा लग रहा था कि कोई क्लास चल रहा हो। तो सर, एक चीज जो लास्ट में मैं आपसे पूछना चाहूंगा कि जो जो आम पब्लिक है उसको अगर नटशेल में अगर समझाना हो कि 2DG क्या करता है या कैसे काम करता है, तो उसके लिए आप उसको हिंदी में कैसे बोलेंगे सर, कि क्या होता है इसका इफेक्ट और कैसे होता है?
तो जैसे जब कोई भी वायरस इन्फेक्शन होता है, तो वायरस इन्फेक्शन में ढेर सारे मेटाबोलाइट्स की रिक्वायरमेंट बढ़ जाती है शरीर में बहुत ज्यादा। तो आम आदमी के रूप में अगर मैं समझाना चाहूँ तो
जी।
इसीलिए कहा जाता है कि आप एक्सरसाइज मत कीजिए उस समय, आप उस समय घर में रेस्ट कीजिए मैक्सिमम। जब वायरस इन्फेक्शन होता है
जी।
डॉक्टर भी आपको सलाह देता है कि आप रेस्ट कीजिए, क्योंकि जब आप एक्सरसाइज करते हैं तो क्या होता है कि ये ढेर सारे मेटाबोलाइट्स आपके ब्लड में आते हैं, और ब्लड से ये मुख्यतः, जैसा कि मैंने बताया कि GLUT3 का एक्सप्रेशन बढ़ जाता है। GLUT3 जो है, जो बहुत मिनिमल भी अगर उसको ग्लूकोज मिलेगा तो उसको खींच के वो वायरस सेल को ले जाकर रिजर्व करेगा, उसके लिए आपकी बाकी कोशिकाओं को अवेलेबल नहीं होगा। और इसीलिए जो जिन पेशेंट्स को ये वायरल इन्फेक्शन हो जाता है वो बहुत टायर्ड फील करते हैं, उनको लगता है एनर्जी नहीं है, क्योंकि उनके बाकी सेल की रिक्वायरमेंट के बजाय वायरस सेल सारा ग्लूकोज और ये सारा खींच लेती हैं। तो अगर क्या नाम है 2DG एक ऐसी दवा है जो देने पे उन्हीं कोशिकाओं में जा रही है, बाकी सामान्य कोशिकाओं में नहीं जाती, तो इसलिए साइड इफेक्ट नहीं हो रहा है इस ड्रग का बहुत ज्यादा, और ये वायरस इन्फेक्टेड सेल्स में जाके एक्युमुलेट हो रही है, उस वायरस को मल्टीप्लाई नहीं होने दे रही है। जैसे अगर हम सामान्य भाषा में कहें कि तो जब दो आर्मी लड़ती हैं तो लड़ने के बजाय किसी भी आर्मी का ये उद्देश्य होता है कि दूसरे आर्मी की राशन सप्लाई को कट कर दे। अगर दूसरे आर्मी की राशन सप्लाई को, इसीलिए आप देखते होंगे कि जो ब्रिजेस हैं और उन सब का बहुत प्रोटेक्शन बढ़ जाता है आर्मी का जब
जी।
वार चल रही होती है, तो ताकि ब्रिजेस नहीं तोड़े जा सके, ताकि आर्मी की राशन सप्लाई फुल लेवल पे मेंटेन हो। सो ये जो टाइम है, इस टाइम में क्या होता है कि वायरस एक आर्मी है जो एक्सटर्नल आर्मी आपके बॉडी में अटैक कर दी है, और आपका इम्यून सिस्टम है जो आपकी इंटरनल आर्मी है, और इन दोनों आर्मी के बीच में वार चल रही है। तो जो आपकी इंटरनल आर्मी है उसको बेसिकली प्रोटीन चाहिए बहुत ज्यादा मात्रा में, क्योंकि सारी एंटीबॉडी और जो ये है उसको प्रोटीन अगर सप्लाई करते रहेंगे आप, तो वो एंटीबॉडी और इम्यून सिस्टम को मजबूत करेगा। लेकिन आपका जो कार्बोहाइड्रेट का कंजम्पशन है, आपका जो फैट का कंजम्पशन है, वो बहुत मिनिमल लेवल पे चले जाना चाहिए। मैं इस चीज को कोरिलेट करूंगा, एक बहुत पुरानी प्रथा है भारत में। आप सभी जानते हैं कि जब चेचक निकलता था, चेचक एक वायरल डिजीज है, स्मॉल पॉक्स, तो उस टाइम में गांव में कहा जाता था कि देवी आ गई हैं, और देवी को तेल पसंद नहीं है, छौंकना पसंद नहीं है, तो इसलिए उन घरों में जो भोजन बनता था वो बिना ऑयल के बनता था, मतलब जो सब्जी भी बनती थी वो बिना ऑयल के बनती थी। तो मुझे लगता है कि उसका कारण शायद यही रहा होगा कि आपका फैट कंजम्पशन जो है वो सिग्निफिकेंटली लो होना चाहिए, सिग्निफिकेंटली लो या बिल्कुल नहीं होना चाहिए फैट कंजम्पशन। क्योंकि वायरस आप सभी जानते हैं, हमने इतना ढेर सारा सैनिटाइजेशन और जो इतना हाथ धोया है डिटर्जेंट से कोविड में, वो इसीलिए धोया है कि जो लिपिड से बना हुआ वायरस है वो डिटर्जेंट डिटर्जेंट में घुल जाएगा।
जी
तो लिपिड से बना हुआ है वायरस ये, और उसको लिपिड की रिक्वायरमेंट बहुत ज्यादा है। तो ये मेटाबॉलिज्म पर्सपेक्टिव से आपका जो कार्बोहाइड्रेट और लिपिड का कंजम्पशन है, फैट का, वो मिनिमम होना चाहिए और प्रोटीन का कंजम्पशन ज्यादा होना चाहिए। और अगर आप इन्फेक्ट, तो इससे आप प्रिवेंटिव रह पाएंगे। और फास्टिंग भी इसमें हेल्प करती है, तो इससे आप प्रिवेंट कर पाएंगे। और जब आप इन्फेक्शन हो जाता है तो आप 2DG लीजिए, जो उसके मेटाबॉलिज्म को जाके वहां इनहिबिट कर देगा, तो उसकी वजह से वायरस मल्टीप्लाई नहीं कर पाएगा आपके शरीर में। तो इस तरीके से हम पूर्ण रूप से इस बीमारी से बच सकते हैं।
थैंक यू सर, बहुत अच्छे से आपने एक्सप्लेन किया और आज हमारे बीच आए और हम लोगों का थोड़ा इनलाइटनमेंट किया कि ये 2DG कैसे काम करता है, और बहुत ही अच्छे तरह से आपने बताया है। तो कोई प्रश्न वैसा मेरे पास अभी तो नहीं है जो मैं पूछ सकूं, लेकिन आपको धन्यवाद देना चाहूंगा कि आप अपने जो बिजी शेड्यूल आपका चल रहा है, मैं कोशिश कर रहा था आपसे कनेक्ट करने का बीच में, और काफी बिजी रहा था आपका शेड्यूल, लेकिन अंततः आपने समय निकाला और आए, और आम जो पब्लिक है उनके लिए आपने ये लेक्चर दिया। इसके लिए बहुत बहुत धन्यवाद सर। नमस्कार।
थैंक्स रंजीत, नमस्कार।
थैंक्स।